Welcome to the Deep Dive, where we take a massive stack of articles, research, and technical notes and just pull out the essential knowledge you need.
And today we're doing a really high-stakes deep dive.
We're looking into biomaterials. Specifically, that grueling, really complex transition from amazing lab science, stuff made from biology, into a core industrial input.
And when we say high stakes, we really mean it in economic terms.
Oh.
The market for these bio-derived materials and ingredients, it's monumental.
Monumental how? What are we talking about?
Well, our sources project this whole sector is going to grow from around 190 to 200 billion dollars today to more than 500 billion by the early 2030s.
$500 billion. That's a massive shift in how we build basically everything. Car parts, clothes, food packaging.
Absolutely. And that growth is powered by really two main categories, advanced polymeric and composite materials, and then these high-purity bio-derived ingredients, think specialized fats or proteins.
And what's so fascinating is that this shift, it's not just a feel-good marketing story anymore. It feels like it's being driven by these, immediate, almost existential pressures on big brands.
It's a combination of stick and carrot, mostly stick, though.
Right.
First, you've got these escalating regulatory drivers. We're seeing new, really aggressive rules, especially somewhere like the EU, targeting single-use plastics. They're implementing carbon pricing. They're demanding just radical ingredient transparency.
So if you don't adapt, you just lose access to major markets.
You're locked out. And then there's the economics. The volatility in petrochemical prices over the last few years has made sourcing bio-based inputs look like a necessary hedge.
A hedge against supply chain shocks.
Precisely. And
okay, so let's unpack this. Because this context, it brings us to the core conflict we saw across all the source material. The big $500 billion question isn't really about the science anymore, is it? It's not, can we engineer a great strain in a perfect little flask?
No, not at all. The question today is entirely about endurance.
Right.
It's, can that material, that process, that meticulously engineered strain, can it survive the messy reality of variable feedstocks, industrial machinery, complex regulators, and most importantly, investors who want actual financial returns?
Not just a deck full of nice ESG slides.
Exactly. Not just pretty graphs. Investors are past that enthusiasm phase. They want de-risked manufacturing.
Which is why we need to start with a warning, right? For any founder or researcher listening.
A really explicit warning. Many, many highly promising biomaterial ventures, backed by excellent science, fail completely. And they fail because of this structural disconnect between the lab and the factory.
So where does that great science usually break down? Is it always the same set of problems?
It tends to break down at four pretty consistent points, creating these four critical gaps. So first, you have the research team's obsession with laboratory optimization.
Meaning they're just focused on the numbers in the lab?
Right. Maximizing titers, getting a perfect tensile strength in an ideal test setting, or collecting clean in vitro data.
It's necessary science, but it's totally isolated from reality.
And that isolation, that's the second gap. Manufacturability at scale. Moving from a little 5-liter bioreactor to a 50,000-liter tank, it just introduces chaos.
What kind of chaos?
All of a sudden, you're battling real physical things like oxygen transfer limitations, massive mixing challenges, and biological fouling, which, basically means the organisms gunk up the tanks.
And that means reject rates, I assume.
Reject rates that suddenly balloon your costs. And then, once you've fought the physics of the factory, you hit the paperwork.
The third gap. The regulatory and quality system hurdle.
Exactly. Establishing these rigorous, traceable quality systems that major brands demand. And getting the right classification, whether it's GRAS approval for food or a specific material certification.
And the final gap is probably the hardest for a startup.
It is investor expectations. Biotech timelines are long. They're capital intensive. Investors want proof that your material isn't just a discovery. It's a factory-ready product with a clear cost path.
A path that justifies sinking hundreds of millions into construction.
That's the bar.
This sounds incredibly difficult. So how do the successful teams do it? The sources we read drew a really clear line between two opposing philosophies here.
Right, we can call them technology first versus application-centric.
Okay, so the technology first approach, that's the risky, classic, science-driven path, right? This is when a founder says, we've got this fantastic new polymer, now we'll go look for a market that needs it.
The hammer looking for a nail.
Exactly.
And that so often leads to years of just searching for a market that can tolerate their material's weird performance profile, its cost, its limitations.
But isn't that path sometimes necessary for a real breakthrough? And doesn't technology first attract venture capital faster because it just sounds so innovative?
It does attract that initial excitement, but the data really shows it leads to a much higher failure rate later on. The successful path, the application-centric path, it flicks the script entirely.
How so?
They start not with the science, but with a highly specific, defined market need. They ask, okay, a major company needs a heat-sealable film for their existing form-fill-seal lines that cost less than X.
Can you explain form-fill-seal lines for us?
Oh, sure. That's just industry jargon for the high-speed machines that take a flat roll of film, form it into a bag, fill it with something like potato chips, and then heat seal it shut.
Ah, okay.
And if your new biofilm doesn't seal reliably at, say, 150 pouches a minute, the line stops, and the company rejects your product instantly. Doesn't matter how sustainable it is.
The stakes are incredibly concrete. So these application-centric teams, they work backward, from the factory, not the biology.
Completely backward. Before they even engineer the strain, they define a huge checklist. They define a specific product archetype, not just a plastic substitute, but a monomaterial flexible film that runs on our customers' current hardware.
And then they get even more specific.
Oh, yeah. Then they drill into what's called the machinability windows.
Which we...
This is critical. They define the exact sealing temperature range the material has to handle. They define the required coefficient of friction, which dictates how smoothly the film slides over the rollers, and they figure out the allowable curl, how much it can warp at high speeds without breaking.
So they're literally designing the biology to fit the performance specs of a 20-year-old machine.
That's exactly it. And beyond the machine, they nail the material specs: oxygen barrier, water vapor transmission, impact resistance. And if it's an ingredient, they define its formulation compatibility.
Meaning what pH, what salts, what other stuff it has to survive when it gets mixed into the final recipe.
Yes. The lesson is just undeniable. Starting your development based on line speeds, shelf life, and regulatory class is so much less risky than starting with a scientific discovery and just hoping to find a home for it.
That brings us straight to the contrast between lab metrics and real-world metrics. This seems to be the point where ventures either succeed or, well, often just derail completely.
It is. We can start with what researchers are trained to optimize. They aim for these perfect results: high titers, strong yields, all in this neat, ideal media, clean growing conditions.
They're measuring things like tensile strength using these perfect standardized test coupons in a controlled lab.
Right, that's the classic method. But the industry, the customer, the investor, they look at completely different indicators. They care about reject rates, they care about web breaks, and they care about downtime on their commercial lines.
Web breaks. That's when the continuous sheet of material, the web, just snaps under tension because it's running too fast or it's inconsistent.
Exactly. And if your beautiful biofilm snaps every half hour, that line is stopped. It's costing thousands of dollars a minute in lost production, wasted labor.
So the cost of failure is the real metric.
That's the only metric that matters at that point. They also care deeply about batch-to-batch consistency, making sure the molecular weight or the crystallinity of your polymer doesn't fluctuate from one batch to the next.
And for something like a probiotic, you have to prove that it's still viable, still active, and that the sensory profile, does it taste weird, is stable in the final product.
Yes, after it has been through high-temperature pasteurization, extreme shear, and 6 months on a shelf, not just in the lab.
This leads to a pretty hard truth, then.
It does. And this is the line that has to resonate with every founder and scientist listening. If your data package is 90% lab analytics titers, perfect coupons, in vitro data, and only 10% real-world performance data,
like line trial stability tests,
then you do not yet have a product. You have a research project.
Let's make this even clearer with some examples of where this industrial reality really bites. Let's start with a bio-based foam material.
Okay, so imagine a company developing a PLA-based foam, maybe with some nanocellulose additives. It's designed to be a lower carbon replacement for, say, styrofoam packaging.
And the academic studies are incredible.
They're amazing. The formula matches or even outperforms conventional foams on insulation, on compressive strength.
Sounds like an easy win for sustainability.
You'd think so. But the commercial relevance hinges entirely on processability. It has to be compatible with existing high-speed foaming and molding lines machines designed for petrochemicals with acceptable cycle times.
So the breakthrough isn't the formula, it's getting it to behave in the machine.
It's achieving stable, uniform foaming behavior and consistent cell morphology when you subject it to the intense heat and pressure cycles of a massive industrial machine. If the cell structure collapses or it shrinks or the cycle time triples,
It's prohibitively expensive.
the factory only cares about the cost of the finished, dimensionally stable part, not the neat recipe.
Okay, let's shift gears. A bio-based platform chemical. This is the drop-in replacement, scaled up through fermentation, feeding into things like polyesters,
Right. And in this scenario, the investment criteria are incredibly strict because it's a commodity. This chemical has to be truly drop-in.
meaning the exact same molecule, same performance, no risk for the buyer.
Zero performance risk. The heavy lifting for these successful ventures is therefore done entirely outside the core biology lab. It's all in process intensification, making the output dense and efficient and massive scale-up engineering.
So they're de-risking the supply chain and the manufacturing, not the molecule itself.
Precisely. The focus is no longer on endlessly tweaking a microbial strain for another 0.5% yield improvement. It's about making the entire refinery operationally sound and financially predictable.
Okay, our third example, nanocellulose packaging film. We know the lab data here could be amazing. Why does this so often fail at scale?
This is the ultimate example of the lab plot versus line scrap problem.
Right.
The first industrial trial often results in a massive double-digit reject rate. And it's not because the barrier property failed, it's because of web instability.
The film is just flapping, tearing, or stretching unevenly as it flies through the machine at high speed.
Exactly. It can't maintain tension, it sticks to the rollers, it seals unpredictably. All those amazing lab plots showing low oxygen transfer become instantly irrelevant.
Because the production line is stopped and the pile of wasted material, the scrap is huge.
That scrap rate determines the viability. The technology's commercial readiness level is determined more by those waste rates than by the material strength curves. And until a material shows stable machinability and low scrap,
Investors and brands will treat it as high risk.
no matter how elegant the science is.
This gives us really specific guidance then for the founders and researchers trying to navigate this. For startup enthusiasts, the takeaway seems to be about relentless specificity.
Absolutely. Define your very first product in concrete operational terms. Specify the customer's line type, the exact format, a pouch, a bottle, the required speed in units per minute, the target price, the regulatory class. You build your strain and your material to fit that reality.
Not the other way around.
That-
And you have to make factory trials your central milestone, not just a final check.
Exactly. Pilot scale line trials, real formulation stability tests. That's the hard data that de-risks your story for customers and investors. That data package is your currency.
And for the scientists working in this space, we have a clear request to broaden your experimental design.
Yes. Design your experiments to mirror real-world process conditions. Incorporate shear stress, temperature cycles, wide pH ranges, and critically, use real-world matrices. Don't test your ingredient in neat water. Test it in the acidic finished beverage it will actually enter.
And I love this piece of advice. Add one factory slide to every paper or talk you give.
Yes. Show how your material behaves in at least one realistic application scenario. A photo of it running on a converting line, a graph showing its consistency over 50 batches. That's the translation step that shows you understand commercial viability.
So let's synthesize all of this. What's
Yeah.
the final thought?
The future of the biomaterials field, it's not just going to be shaped by the smartest biology or the most elegant chemistry. It's going to be shaped by the teams that can fluently speak both languages. The language of titers and tensile curves, and the absolutely essential language of lineup time, double-digit scrap rates, and regulatory approvals.
And they have to connect all of that to a product a consumer can actually hold in their hand.
And use reliably. That's the goal.
Fantastic, actionable insight. We focus so much today on how new biology must conform to existing industrial infrastructure. The machines, the processes already in place. So I'll leave you with a final thought to mull over. Given this reliance on existing equipment, how often is a true breakthrough in industrial process engineering, developing an entirely new machine or a radically more scalable reactor, actually more valuable and harder to achieve than a breakthrough in chemistry? Something to consider as you watch this $500 billion market restructure itself. Thanks for diving deep with us.